This is the second half of the presentation for hematopoiesis. Now we'll talk about the maturation characteristics of blood cells. Well, one, we should understand that blood cells has to undergo maturation stages in the bone marrow before they are released in the blood during their maturity so that they may perform their function. In any cells series, a progression of cells exists between the mature and the most immature blast cells and the mature cells. So that means there are stages in between. Sometimes it is really difficult to identify the stage, okay, by looking at them under the microscope. The general rule is to identify the cell that has the most mature form. So how do we identify these cells? First, of course, we need to use a right stain smear and take note of the size of the cell, the nuclear cytoplasmic ratio, the nuclear characteristics. This includes the chromatin pattern, the nuclear shape, the presence of the nucleoli, and the cytoplasmic characteristics, including the color, the granulation, the vacuoles, and the shape of the cell. So, for what exactly happens during the maturation? Number one, the size of the cell decreases. The cytoplasmic and the nuclear, or the nuclear and cytoplasmic ratio decreases from 4 is to 1 or 3 is to 1 to 2 is to 1 or 1 is to 1. So that means that the size of the nucleus becomes smaller related to the size of the cytoplasm. There are exceptions which include the erythrocytes and the thrombocytes because they don't have nuclei. Small lymphocytes also are excluded because most of the time they retain the original ratio. The nuclear characteristics, the changes in the nucleus include, number one, the chromatin pattern becomes more coarse and dense. So initially it is fine, granular, then later it becomes coarse. Then the nuclear shape changes from unlobulated to multilobulated or segmented. And another noticeable change is the disappearance of the nucleolus. In the cytoplasm, we should be able to observe a change in the color from deep blue, okay, to lighter blue in lymphocytes, bluish gray in monocytes, and or pinkish in case of the red blood cells. Another characteristic that is notable is the granulation. So in this case, specific granules appear later in the development of granulocytes. Another important characteristic that we should note of is the vacuolation. So that means the presence of vacuole, which increases as the white cells mature except for monocytes, in which case they frequently have vacuoles throughout their life cycle. The shape of the cell, especially in the megakaryocyte, okay, as it assumes an irregular outline. So in order to identify the cells, we have to examine them more systematically by assessing the various maturation features. Now for the blast, so meaning they are young cells. So in the development of these blood cells, the earliest identifiable cell is called the blast cell. So it is large. It has a rounded nucleus with fine chromatin threads and nucleoli. So another thing is that the cytoplasm is small in amount and usually dark blue in color. That means it is basophilic. So the size of the cell is about 10 to 20 micrometers in diameter with a high nuclear to cytoplasmic ratio. So if you remember, it's about 4:1. Okay, so that means that the nucleus is 4 times bigger, or the amount of space occupied by the nucleus is 4 times compared to that of the cytoplasm. Additional tests are required to identify the plasma, especially in malignancies like leukemia. So the nucleoli depends on the type of cell. For example, for myeloblasts, it may have about 1 to 5 nucleoli. For lymphoblasts, about 1 to 2 nucleoli. For monoblasts, about 1 to 2 nucleoli, but sometimes it may be 3 to 4. For erythroblasts, it may have up to 2, and the color may stain darker compared to the other blast cells. For megakaryoblast, they may have 1 to 5 nucleoli, so that's similar to myeloblast. So, production of specific cell lines. Number 1, production of the erythrocytes, or what we call erythropoiesis. So, this term means the production of red blood cells. It starts in the primitive erythrocytes in the embryonic yolk sac. So, this is in embryo. And there are necessary substances that are should be present for the erythrocytes to be produced, like amino acids, iron, vitamin B12, B6, and folic acid. These are B vitamins, and trace minerals like cobalt and nickel. So the hormone that regulates the production of red blood cells called erythropoietin, which is produced by the kidneys in response to hypoxia. 10 to 15% of erythropoietin may also occur in the liver. Take note. Androgen and thyroid hormones may also stimulate the action of red blood cells. Erythroid precursors, that means the younger form of red blood cells, are derived from the CFU GMM, and the earliest progenitor committed exclusively to this red blood cell lineage is the burst forming unit erythroid, or the BFU-E. This stage is then followed by the CFU-E, or the colony forming unit erythroid. And the earliest recognizable RBC precursor is called the proerythroblast, which has fine nuclear chromatin and intense blue cytoplasm. The next stage is called the rubriblast. So this is again the pronormoblast, also known as pronormoblast. The size is about 20 to 25 micrometers in diameter. So this is like more than twice the size of an adult red blood cell. The nucleus is large, round to oval, and it contains 0 to 2 light bluish indistinct nucleoli. The chromatin forms a delicate network giving a nucleus of a reticular appearance. So that means that there may be some form of meshwork inside the nucleus. The cytoplasm is narrow, about 2 micrometers, rim of dark marine blue cytoplasm with a perinuclear halo. So, and the ratio is about 4:1. The next stage is called the basophilic normoblast, or the prorubrocyte. The size is much smaller, about 16 to 18 micrometers in diameter. The nucleus is round or oval and smaller than the previous stage. The chromatin forms a delicate clumps, so that means that they are become more granular, so that its pattern appears to be denser and coarser than the pronormoblast. No nucleolus or nucleoli are present. Cytoplasm is slightly wider, ring of deep blue color. Okay, compared to the normoblasts, there may be a perinuclear halo. That means that there is something like a ring, okay, surrounding the nucleus. And the nuclear to cytoplasm ratio is also the same, 4:1. Next, prochromatophilic normoblasts, also known as progresite. So the size is smaller, 12 to 14 micrometers in diameter. The nucleus is also smaller than the previous stage and has a thick membrane. The chromatin pattern is coarse and appears in masses. Cytoplasm, the nucleus is shrinking. The band of the cytoplasm is therefore widening. The tint of the cytoplasm is described as polychromatophilic or polychromatic, meaning it has a combination of blue and red color. Okay, so we describe that as lilac. The ratio may vary from 2:1 to 4:1. Orthochromatic normoblast, the next stage. The size is almost that of a mature erythrocyte, 10 to 12 micrometers, small and central eccentric nucleus with homogeneous structureless chromatin. Okay, and the nucleus is extruded or removed by the cell. Cytoplasm is wider, okay, because of the shrinkage of the nucleus. The entire cell is somewhat smaller than the polychromatophilic normoblast. The ratio of the nucleus and cytoplasm is now 1:2 to 1:3. So the nucleus is now very much smaller than the cytoplasm. Reticulocyte. So this is the stage before the mature form of red blood cell. So this is more basophilic and nuclear, meaning there is no more nucleus. It's already extruded or expelled from the nucleus, from the cytoplasm. The remnants of the RNA may be seen if you use a special stain. Okay, and this is the reason why we term it reticulocytes, the presence of some form of reticulum, which are remnants of the RNA. Okay, so these are filamentous structures in chains or single dots. Okay, if you stain it with new methylene blue. In right stain, they may appear as large bluish red cell with polychromatophilic macrocytes, so macrocytic. That means they are larger than normal. The network is responsible for the name, as I have mentioned. Next, as the bone marrow reticulocyte matures, the network becomes smaller, finer, thinner, and finally it disappears in 3 days' time. About 1% of reticulocyte may be present in the peripheral circulation, so this is normal. So the size is about 8 to 10 micrometers in diameter. So this is already the size of a mature red blood cell. The nucleus is absent, and the cytoplasm appears more bluish or basophilic, faintly bluish or basophilic. The mature erythrocyte. So the size is about 7 to 8 micrometers in diameter, still much smaller than the reticulocyte. The cytoplasm is described as biconcave disc shape that is filled with hemoglobin that allows it to assume an orange-pink color, okay, with a pale staining center, okay, described as central pallor. Next. So these are the stages in summary in table form. You start off with proerythroblast followed by basophilic erythroblast, polychromatophilic erythroblast, orthochromatophilic erythroblast. So as you can see, they are just describing the color of the cytoplasm Okay, and until finally it becomes a reticulocyte, then a mature erythrocyte. Notice the change in the size of the nucleus, the size of the cell, the what else, the ratio between the nucleus and the cytoplasm, the presence or the absence of the nucleus, and the condensation of the chromatin bodies. So those are the things that we need to take note of in describing and comparing the different stages of the erythrocytes. Next, regulation of erythropoiesis. We already know that there are some hormones that can trigger the production of red blood cells in the bone marrow. So primarily it is the erythropoietin, which is a glycosylated hormone, about 40% carbohydrates with about 165 amino acids, polypeptide. So normally 90% of this hormone is done or produced by the kidneys, particularly the juxtaglomerular part of the kidney or complex, and 10% is also produced by the liver and other organs, okay, other places. So there is no preformed stores of erythropoietin. So that means it is freshly produced, and stimulus is uh, when there is low oxygen tension in the tissues, particularly the kidneys. So production is increased when there is hypoxia. So this may be due to anemia, when there is low hemoglobin level, when there is impairment in the oxygen release from hemoglobin. So if there is a problem with hemoglobin, and poor blood flow. So in cases of circulatory problems, okay. So for example, if the heart is unable to pump blood properly, so it may not reach certain tissues. And low atmospheric oxygen in case you are located in a high altitude places like the mountains. So the production of this erythropoietin will stimulate the process of erythropoiesis, and there will be an increase in the number of progenitor cells committed to erythropoiesis. So we know that is BFU-E, then later CFU-E. Okay, erythropoietin accelerates every stage of the red cell production. So all of those stages is accelerated. It increases the rate at which those committed stem cells divide and differentiate. So there will be more stem cells produced, okay, and increase the rate of cell division. It speeds up the incorporation of iron into the developing red blood cells. As you know, iron is a necessary mineral in the development of red blood cell. It shortens the time of cell maturation, so it becomes faster. It hastens the entry of reticulocytes in the peripheral circulation. So you might find that there is an increase in the number of reticulocytes in peripheral circulation. So, on the other hand, reduced erythropoietin activity can also happen if there is an increase in the amount of red blood cell, like in polycythemia vera, and when the ability of the oxygen, hemoglobin to release oxygen to the tissue is more than. This is slide 77, ineffective erythropoiesis or intramedullary hemolysis. Erythropoiesis is not entirely efficient because 10 to 15% of that is ineffective in a normal bone marrow. The developing erythroblasts will die within the bone marrow without becoming mature cells. So together with their hemoglobin, this will be ingested by the macrophages. And an increase in this process may result in anemia. Okay, example, megaloblastic erythropoiesis. So megaloblasts are large, okay? They are pathologic cells that are not present in the normal adult bone marrow, So the appearance of this type of erythrocyte is a result of deficiency in either vitamin B12 or folic acid or both. So you should recall that vitamin B12 is normally found in meat products. So that means if a person is a vegan, he or she may not have enough intake of vitamin B12, okay, or folate, okay. And also another thing about this vitamin B12 is that it is produced by the bacteria in our gut. Deficiency of the vitamins may lead to defective DNA synthesis, leading to megaloblastic erythropoiesis. So the nucleus, the cytoplasm do not mature at the same rate, okay, and the nuclear maturation lags behind because of cytoplasmic hemoglobinization. The nuclear lag appears to be because of interference with the synthesis of the DNA, but RNA and protein synthesis continue as normal. The end stage of megaloblastic maturation is called the megalocyte. So this is a large RBC with a diameter of about 9 to 12 micrometers. So there are some cases of parasitic infection that leads to megaloblastic anemia, like in case of fish tapeworm infection. Okay, now let's talk about the process of formation of the WBCs, also called leukopoiesis. 1. Granulopoiesis and monocytopoiesis. So that means the production of granulocytes and monocytes. Neutrophils and monocytes arise from a common progenitor. That's why we are taking this up together. So the myeloblast is this earliest precursor. Say on the division of the myeloblast, it gives rise to the promyelocyte. So that means that's the second stage. Then from this promyelocyte is described as having an abundant dark azurophilic primary granules that overlay the nucleus and the cytoplasm. So that means it's well distributed. So azurophilic, so from the word azul, okay. So that means it's more like bluish, but it's not entirely blue. So with subsequent cell divisions, these primary granules later become diluted with secondary granules. So we call this the specific granules or neutrophilic granules. So the concomitant cell division and maturation sequences continue to form promyelocytes to become early myelocytes, then late myelocytes, then metamyelocytes. So in this case, the metamyelocyte will mature and the nucleus will become more attenuated, and the cell will now be called a band or the stub form because of the it will assume a dumbbell shaped, okay, parang dumbbell shaped nucleus because it's like bent, okay, forming equal sides. Subsequent segmentation, so it will proceed to more segmentation of the nucleus, will give rise to a mature neutrophil or a polymorphonuclear leukocyte or PMN. Slide 82, continuation of granulopoiesis. The average interval of the initiation of granulopoiesis to the entry of mature neutrophil into the circulation takes about 10 to 13 days, and the mature neutrophil remains in the circulation for only about 10 to 14 hours before they enter the tissue, where it will die after performing its ability to phagocytize, okay, harmful organisms or cells. Next, so we're now going to describe each of the stages of the maturation series for leukopoiesis. The first one, of course, is the myeloblast. The size is about 10 to 20 micrometers with a round or oval shape. The nucleus is large, oval, or rounded, and eccentric. So that means that is not in the center. It has a thin nuclear membrane, finely dispersed granular purplish pale chromatin with a well-demarcated pink, evenly distributed parachromatin. It has about 2 to 5 light blue-gray nucleoli surrounded by dense chromatin. For the cytoplasm, it is small compared to the nucleus with a nucleus-cytoplasmic ratio of about 7 to 1. You can see here that the nucleus is much bigger compared to the cytoplasm. Okay, the color is basophilic and with a small indistinct paranuclear lighter staining halo because of the Golgi apparatus. The cytoplasm is without granules. Next stage is promyelocytes, second stage. So larger than from the myeloblast. Okay, 15 to 20 micrometers in diameter with the shape being the same, round or oval. The nucleus is large, still it begins to shrink, round or oval, eccentric, possibly slightly indented, and with a thin membrane. There may be the presence of 1 to 3 nucleoli, faintly visible within the chromatin. Okay. Next, the cytoplasm is pale blue, somehow larger than the myeloblast. So the ratio is now about 4 is to 1 or 5 is to 1. The basophilia is not quite as intense as in myeloblast. So that means the dark cytoplasm, that dark bluish cytoplasm becomes lighter in color. Not very light, but lighter compared to the previous stage. Contains dark abundant azurophilic primary granules. Okay, and what else? These non-specific peroxidase containing azurophilic granules are characteristic of this stage, promyelocyte. So it is peroxidase positive. Take note of that. That is one of the stain, okay, enzyme that we use to identify the promyelocyte. Next stage is the myelocyte. Let me adjust. Okay, so myelocyte. This stage is the myelocyte. You can see the image on the upper right corner. Unfortunately, it's not very clear. Anyway, so this is the last stage that is capable of cell division. Take note of that. So after this, there will be no more mitosis. The size is about 10 to 18 micrometers in diameter, rounded, with a nucleus that is described as condensed, oval, slightly indented, and eccentric. The chromatin is coarse and the nucleoli is already absent. The cytoplasm is now showing a light pink color with neutrophilic granules. So this is now the specific granules. So granules may cover the nucleus and are coarse in the younger cells but later become finer as the cell matures. The NC ratio is about 2 is to 1 or 1.5 is to 1. Next stage is the metamyelocyte, slide 88, also described as a juvenile cell. The size is about 10 to 15 micrometers when rounded. The nucleus is eccentric, condensed, indented, or may assume a kidney shape. The nuclear membrane is thick and heavy, and the chromatin is concentrated into an irregular thick and thin areas. Cytoplasm appears abundant, so meaning there is more cytoplasm this time, and assumes a pale or pink color. So the presence of specific and unspecific granules, okay, which vary in sizes, okay, and the color is like a mixture of basophilic and eosinophilic granules. Okay. Ratio of nucleus to cytoplasm is 1 is to 1. Next, band granulocyte or the stub cell. So this is also another juvenile cell, young cell. Okay, and the youngest granulocyte normally found in peripheral blood. So you might find this in the peripheral circulation. The size is about 9 to 15 micrometers in diameter. This is already the size of a mature WBC. Okay, particularly the basophil, eosinophil, and neutrophil. Nucleus is elongated, curved with a U shape. Okay, so that this is why they call it the band or the stub cell. But it may be twisted. So it's not segmented but may be slightly indented at 1 to 2 points. The chromatin is thick and coarse and parachromatin is scanty. Cytoplasm contains specific and unspecific granules. Okay, the color is pink or colorless. The nucleus and cytoplasm ratio is 1:2, more cytoplasm. So segmented granulocytes still, so this is 9 to 12 micrometers in diameter, not 0 to 12, with an eccentric and heavy thick chromatin mass, okay, divided into 2 to 5 lobes connected to each other by thin bridges. They are very thin bridges, so that's why this is described as segmenters. So, and then the ratio of the segmented to band form is about 10:1. So, for every one band, you might see like 10 segmenters. Cytoplasm is abundant with eosinophilic color or even colorless. They have specific granules. That means if this is a neutrophil, so the color of the granules should be neutral. Okay, so that means parang purple color. The neutrophilic granules are very fine textures and do not overlay the nucleus. So that means they are distributed in cytoplasm but not in the nucleus. That is why they do not cover the nucleus. The nucleus and cytoplasm ratio is about 1:2. Next, eosinophilic granulocyte and precursor. So this is a separate series. So eosinophils mature in the same manner as neutrophil. It starts off as a myeloblast. Okay, however, it's not really recognizable. So this eosinophilic promyelocyte, which is the next stage, is found in stained preparation and the granules are described as bluish. Then later, it matures to become more orange in color because it's eosinophilic. So the mature eosinophilic granules are larger than neutrophilic granules. This is now the specific granules, so round or ovoid, so prominent in eosinophilic myelocyte. So that's the stage when you can find these specific granules. I hope that at this point, I just want to mention that there are two types of granules, the primary and the secondary granules. The secondary granules are also known as the specific granules, which are found in specific granulocytes. So now the mature eosinophil. So the size is about 10 to 16 micrometers in diameter, slightly larger than a segmented polymorphonuclear granulocyte, but this is hardly noticeable most of the time. The size of the neutrophil, basophil, and eosinophil is almost similar. Nucleus is eccentric, usually bilobed, take note of that, and rarely single or trilobed, and may contain dense chromatin masses. Eosinophils with more than 2 nuclear lobes are found in cases of deficiencies with vitamin B12 and folic acid. So if you remember, this is found in megaloblastic anemia and in cases of allergic disorders. So, next. The cytoplasm of mature eosinophil is filled with orange-pink specific granules, and the granules are uniform in size. Compared to neutrophil, of course, they are larger, okay, neutrophilic granules. So they are larger. Okay, and they do not cover the nucleus. That is why it is possible to identify, describe the nucleus of eosinophils. So highly metabolic and contains histamine and other substances. Basophilic granulocytes and precursors. So this is another group of WBCs. The early maturation of um, basophilic granulocytes similar to that of the neutrophilic granulocytes. That means we start off with the myeloblast. Okay, so the mature basophil is somewhat smaller than eosinophils with a size of about 10 to 14 micrometers in diameter. So again, sometimes the sizes of this basophil, eosinophil, and neutrophil is hardly noticeable. That's the difference. I mean, is hardly noticeable. Okay, and you may not be able to find this basophil in your lifetime as a medical technologist. You might find this only in books, but not in the actual laboratory setting. So if you find a basophil, you might as well take a picture of it so that it will be your a remembrance of that event. So the nucleus assumes an S-like pattern. However, it's difficult to see the nucleus because of the large granules. So it's masked by the cytoplasmic granules. The cytoplasm is described as blue to pale pink. Okay, again, you may not be able to appreciate this color because of the presence of uh, large numbers of granules that overlie the nucleus. Okay, however, they do not really fill the entire cytoplasm completely, unlike the eosinophils. So there are more granules in eosinophils compared to the basophils. However, the size of the granules of these basophils are much, much larger. Okay, next, the monocyte and their precursor, another group. So, since the monoblasts cannot be differentiated from myeloblasts morphologically or histochemically, so histochemicals, that is the use of enzyme stains. Okay, one may assume that the myeloblasts can also give rise to a myeloid or monocytic cell. So that means we cannot really identify which is a monoblast and which is a myeloblast. Anyway, the size is 15 to 20 micrometers. The nucleus is rounded or oval and at times notched and indented. There is chromatin, which is delicate blue to purple stippling, so it's like dots. They are like dots. Okay, and small regular pink, pale, or blue parachromatin areas. So there are areas which are of different color. The nucleoli, which may range in number from 3 to 5, are pale blue, large, and rounded. The cytoplasm of this monoblast is relatively large in amount, may contain some azurophilic granules. The color is pale blue or gray. The cytoplasm is filled with nuclear indentation. That is filled with the nuclear indentation lighter in color compared to the rest of the other parts of the cytoplasm. So you might find the presence of our body. So our rods, they are rod-like, parang mga rods, rod-like structures in the monoblast. Promonocyte, the next stage, the earliest recognizable stage in the monocytic series is called the promonocyte. Capable of mitotic division, so they can divide. Its product, which is the mature monocyte, is only capable of maturation into macrophage. So it can transform to become a macrophage. Anyway, we have already noted that one of the macrophages in our body is the monocyte, which is in the blood. So the size is about 12 to 20 micrometers in diameter, much bigger than the monoblast. Okay, the nucleus is large, ovoid to round, convoluted. So that means it may be like coiled or twisted, grooved. So, meron tayo mga indentations, indented. Chromatin forms a loose or open network containing a few large clumps. So there may be the presence of two or more nucleoli. The cytoplasm is sparse, very little cytoplasm is present, grayish-blue in color, with fine azurophilic granules. The ratio of nucleus to cytoplasm is 3:1. The size is 12 to 20 micrometers. Eccentric nucleus or sometimes central nucleus, but most of the time it is in the towards one side of the cytoplasm. So it takes different shapes from brainy convolutions to lobulated to S shape. Okay, or often lobulated nucleus. So you can find the presence of you know like structures inside the nucleus. The chromatin network consists of fine, pale, loose linear threads that are found as small areas in their junctions. No nucleolus is present. The overall impression is that a pale nucleus is which is quite variable in shape. So the cytoplasm is abundant, okay, opaque, grayish blue in color with moderate granules. Okay, unevenly stained and may be vacuolated. The ratio to nucleus to cytoplasm is 1:1. Okay, last, lymphopoiesis. So the precursor of the lymphocyte. Okay, we believe that this is it's the multipotential or pluripotential cell. Okay, capable of giving rise to granulocyte erythrocytes and the platelets. Lymphoid precursor cells travel to specific sites, as you know. Some of them will become T lymphocytes, some of them will become B lymphocytes. So they may differentiate into cells capable of either expressing cell-mediated immune response, like in the case of the T cells, and sometimes they may produce immunoglobulins or antibodies. So the influence of the former type of differentiation, so meaning the the B, okay, the B, okay, the T rather, the T is because of the production of a hormone called thymosin, okay, thymosin in the thymus gland. Okay, to continue, slide 103. So there's about 119 slides. This site of formation of lymphocytes with the potential to differentiate to become antibody-producing cells is not identified in humans, but we believe it is in the tonsils or in the bone marrow. Okay, in chickens there is a part called the bursa of Fabricius, and this is the reason why they are called B cells, because of the bursa of Fabricius. So this was the historical, the very first, the very first time that they tried to differentiate between B and T lymphocytes. So B cells ultimately differentiate into morphologically distinct antibody-producing cells, which are called the plasma cells. So they can differentiate, they can become plasma cells. Okay, so the first stage is called the lymphoblast. Okay, the size is 10 to 20 micrometers in diameter. The nucleus is central, rounded, ovoidal in shape. Chromatin has a stippled pattern, so stippling, meaning dots dotted, okay? The nuclear membrane is distinct with one or two pink nucleoli, okay, and they are well outlined, meaning they are conspicuous, very easily seen. What else? Cytoplasm is not granular, sky blue in color, which may have a deep blue border. It forms a thin perinuclear ring. The ratio of nucleus to cytoplasm is 4:1. Next stage, prolymphocyte. The size is 9 to 18 micrometers in diameter. Nucleus is oval, okay, but may assume an indented shape also. It has a faint nucleolus. The chromatin is slightly condensed with a mosaic pattern. Cytoplasm is grayish blue with bluish edges. It may show the presence of azurophilic granules and vacuoles. Lymphocytes are of two types, as we have mentioned. So the morphologic difference lies in the amount of cytoplasm. There is a small lymphocyte and the big or the large lymphocyte. So the smaller one is about 7 to 18 micrometers in diameter. So almost the size, can assume the size of the red blood cell, mature red blood cell, round or oval to kidney-shaped nucleus. Occupies 9/10 of the cell diameter. So that means it occupies almost entirely the entire cell. The chromatin is dense and clumped, and the nucleolus is present but poorly defined. Cytoplasm is basophilic and forms a narrow rim around the nucleus, at times a thin blue line only, okay, with few azurophilic granules. So it's hardly noticeable if there is even the presence of the cytoplasm. The ratio of nucleus cytoplasm is 4:1. Okay. How do we distinguish a small lymphocyte? So when there is clumping of chromatin around the nuclear membrane, this may help to distinguish it from a nucleated red blood cell. So you see sometimes nucleated red blood cell may appear in the circulation. So that means the stage before the reticulocyte, before the removal of the nucleus. Oh, large lymphocyte, the other type. So they are much larger, 9 to 12 micrometers in diameter. Nucleus is dense, oval, or slightly indented, centrally or eccentrically located. Chromatin is dense and clumped. Cytoplasm is more abundant. There is more cytoplasm. The color is gray to bluish, pale blue color. Unevenly stained and streaked at times. Okay, so there may be some streaks or some irregular patterns. So a few azurophilic granules are found in about 30 to 60% of the cells. So they are called the large granular lymphocytes. So this is slide 109, large lymphocyte continued. The nucleus cytoplasm ratio is 1:1. There is large nucleus. The characteristic of this large lymphocyte is that the cytoplasm is abundant, and they have a tendency to form azurophilic granules. Next, the platelets. The formation of platelets called thrombopoiesis. They are derived from the largest cell in the bone marrow called the megakaryocyte, which originates from megakaryoblast. Okay, so the original is megakaryoblast and promegakaryocyte and the megakaryocyte. So the nucleus of this megakaryoblast, the DNA inside the nucleus, are capable of reduplicating without cell division in the process called endomitosis. So something unique about this cell. Thrombopoiesis as a result of this reduplication. So the megakaryocyte may exhibit polyploidy. So meaning there is multiple nucleus inside the cell. The final stage of platelet production occurs when the mature megakaryocyte sends cytoplasmic projections into marrow sinusoids and sheds platelets in the circulation. In short, it results in fragmentation of the platelets. So thrombopoiesis continued. It takes about 5 days for a megakaryoblast to become a mature megakaryocyte. So each megakaryocyte can produce about 1,000 to 8,000 platelets. So if you try to look into the normal value or the reference range for platelets, it's actually the lowest. Okay, about 100,000 to 300,000 platelets in the per cubic mm. So compared to red blood cells, compared to WBC, this is actually a small number. So the platelet survives for about 7 to 10 days in the peripheral circulation. Okay, so what are the stages? The first stage is called the megakaryoblast. So the size is 10 to 30 micrometers in diameter. The cell is smaller than its mature form. So this is the only time that cells become bigger as they mature and then back to small again. The nucleus is single, large, oval, indented with a loose chromatin structure. There is the presence of multilobulated nuclei. Uh, okay, that represents a polyploid. So we already described this polyploid. So, and then several pale blue nucleoli may be present but are difficult to see, and the parachromatin is pink in color. Cytoplasm is scanty, bluish, patchy, irregular ring around the nucleus. So the periphery may show cytoplasmic projections and pseudopodia-like structures. So that's why, anyway. So the immediate perinuclear zone is lighter than the periphery. So promegakaryocyte, the size is about 20 to 50 micrometers in diameter, larger than a megakaryoblast, as you have noted. Okay, so what else? The nucleus is large, indented, polylobulated. Chromatin appears to be heavily stained, and coarse strands. So what else? The total number of nuclei is decreased, and they are more difficult to see than in the blast cell. The chromatin is thin and fine. Cytoplasm is intensely basophilic, filled with increasing number of azurophilic granules that is from the Golgi apparatus towards the periphery. Okay. So the last stage, the granular megakaryocytes. Okay, so majority of the megakaryocytes in the bone marrow aspirates are in the stage 3. Okay, and is characterized by progressive nuclear condensation and indentation. And this is when the platelets have begun to form. The size is about 30 to 100 micrometers, so that's why we consider this a giant cell in diameter. So in the, what else? The cytoplasm is large with polychromatic appearance. Okay, it has a blunt, smooth pseudopodia-like projections, and it contains aggregates of granules surrounded by halos, pale halos. The structures then give rise to platelets at the periphery of the megakaryocyte. So the last stage, the platelets, so the mature platelets. So compared to megakaryocytes, so these are very much smaller. The smallest cell in the bone marrow, in the peripheral circulation, is the platelet. So the size is about 1 to 4 micrometers. There is no nucleus that is present. And I'd like to mention again that we do not really consider the platelet as a true cell because they are, well, merely fragments of cytoplasm of the megakaryocyte. However, since we know that these megakaryocytes are capable of exhibiting polyploidy, they contain its own part of the nucleus. So, in Wright's gems stain, so this is actually a type of staining technique. Uh, Platelets will appear as small, bright azure, rounded, elongated bodies with a delicate granular structure. Okay, in the so-called CBC, platelet count is not considered part of that. It's a special test. I just want to mention again, I think I mentioned this before. Alright, so thank you very much for listening. I hope you have, absorbed some information, new information during this recorded presentation. If you want to listen again, so you may do so. Thank you. Bye-bye.